it really has been a great meeting. And I really thank all of the organizers and also for those of us who are speakers, all of the emails we've gotten from Sarah and Zoe and all of the meeting staff. It's really coming here, you can just relax and actually enjoy science. And I personally needed that, so thank you. I'm gonna kind of go through and talk about just human skin microbiome and the studies we've done, which for me is maybe even just history of the field of like, you know, what we started, what were the challenges, how did we overcome them, where do we see the field going now? Recognizing that the gut microbiome is probably where most of the action is, but sort of using the skin as an example of the other ways in which the microbiome is the interface between the human body and the environment. So the human body is this ecosystem, and I'm going to talk a lot about the bacteria. We have, I'll talk just a little bit about the fungi and then tell you about the future work we're doing there. I'm going to talk a lot about the viruses and really just the interplay, because I come at this field from genomics. I actually did my PhD in the original MIT Genome Center. It's been really fun for me to watch, to see the history exhibit around here and the origins of the Human Genome Project. And I really thought when I started my PhD that we would find genes responsible for disease. And I actually thought it was going to go hand in hand. We would then be able to cure these diseases. And some of that is happening. I think that there's increased excitement about the concept of gene therapy with new technologies coming online. But a lot of it has ended up being these sort of chronic diseases that are really about what we call gene-environment interactions. But in that, I think a lot of what we end up talking about, there's the toxicology and so on, but a lot of gene-environment interactions end up being the microbes, and the microbes that colonize us, that then are maybe driving disease, maybe exacerbating. And, you know, one of the things that I think has really appealed to all of us is this idea that a lot of these disorders, you know, that famous paper from Bach et al. In New England Journal in 2012 about the rise of these sort of autoimmune diseases. And what this is happening not because our genes are being selected, that you're seeing this rise in autoimmune diseases and asthma, autoinflammatory diseases, but possibly because of the microbes that colonize us. And so I think that's really becoming an essential part of how we understand human disease. So we focus in my group at the NIH on the skin microbiome, and we're really a team. The clinical studies are all led by Dr. Heidi Kong. We initially started with Dr. Maria Turner as well. She's now retired. And really, what we're trying to take advantage of is the topography of human skin. I mean, when you study the gut, and we just heard this, you know, we've heard several lovely talks from Laurent earlier this morning about how, you know, we're measuring the stool, but perhaps it's something happening in the ileum or the, you know, small intestine, and what are we really measuring? Well, the skin, we do have access to the entire tissue, and I think it's probably the one that is, in many ways, most accessible to us, because really no one minds if you swab 18 different parts of their body. And that's in contrast with if, you know, the stool sample would be like as if I sat in a bathtub and at the end of it you know, took the scum off the top and said, this is the skin microbiome. No, we get at it with this dissection, and dermatologists are used to thinking about the topography of human skin. Eczema is going to happen here, psoriasis is going to happen here. And that's all part of the clinical diagnosis when you walk in and you have a rash. It's about your age, you know, but particularly where is this body site? And so for dermatologists, this has made sense for a long time, that there might be some differences in the skin, but also in the microbial colonization. And so we picked these 18 body sites for our study because these are the sites of disease predilection. and we're really trying to run the gamut of what kind of disorders could we see and develop these data sets. So our first studies, and I'll go through this very quickly, were with amplicon sequencing. And we were really just trying to say, is it, are we going to find out new things with sequencing? And what we found here was this topography of the human skin. That basically the difference between body sites of the same individual, that the body site was dominant over the individual. So you can see that here, where I'm showing you like the back is dominated by Propionibacterium acnes. So that's a sebaceous site. And a moist crease is going to have more of the Proteobacteria and the Actinobacterium. Some sites like the inside of the nose, or in particular the umbilicus or the belly button, could be quite distinct between individuals. But basically the bottom line here is that the bend of my right elbow is most similar to the bend of my left elbow, and after that the bend of my left elbow is most similar to Kath's, and more similar than it would be to my underarm, because this is a sweaty site and this is a moist crease. We did the same kind of analyses with fungi using the ITS1 sequencing, and here we found less diversity. It's mostly Malassezia. But we did see differences at the species level, and all of the diversity that we're seeing of fungi is on the feet, which is, again, not surprising in that clinically what we're seeing is that the fungal association is with athlete's foot, and with toenail infections. Those are really what you would think of. I mean, some dandruff. Okay. So I really want to spend the majority of my time today talking about shotgun metagenomics. And I do acknowledge that this is resource intensive and analysis intensive, but I'll at least take you through 2 stories today of how we're using shotgun metagenomics to get to another level of resolution, and what are the challenges that we overcome. And so really, just to say that all of our samples are taken where we swab the skin, take that swab, put it directly into lysis buffer, and make DNA. And we have that same DNA prep no matter what we're going to do with the sample. And if we're going to pursue the shotgun metagenomics, we take that DNA and we make a Nextera library straight from the DNA. And then we're sequencing on the Illumina HiSeqs. We've actually moved on to the X10 NovaSeq instrument. So we're getting about 100 million reads from each sample. What are we doing with this? Well, the first thing that we wanted to know was the relative kingdom abundances, because we're really interested in all 3 kingdoms and moving this field to start thinking about bacteria, fungi, and viruses, and in particular looking at interdomain interactions. I'm going to tell about strain differentiating markers, and I'll sort of explain how skin is not like the gut in terms of our analysis pipelines. And we've had really nice talks on antibiotic resistance and functional characterization. We'll go through a little bit of that too. Okay, so here's our sort of pipeline where from the shotgun metagenomics, we actually mask out the human reads. We can look at those. We do consent the patients for whole genome sequencing, and we have looked at them in terms of identifying mutations in the filaggrin gene or other sort of genes like that. You can actually get that pretty easily out of the human reads. So we take the non-human reads and we're going to do this multi-kingdom analysis where we're looking at bacteria and the different genera, on the fungi and the viruses, and not entirely as elegantly as Trevor's lab, but we, at the same time as we do this microbiome sequencing, we also do culturing, and we find that this coordinated process is really accelerates our discovery because we then have the exact strain from the match to the sequencing. So we develop a similar profile where you can get the bacterial abundance, the fungal abundance, and now for the first time with shotgun metagenomics, you can also see the relative abundances of the kingdoms. So for example, for the toe web, which I told you had a lot of fungal diversity, but now you can see that it's actually mostly bacteria and very little fungi on the feet. And maybe, maybe it doesn't matter. Maybe, they can have large effects. But that also can help you then when we do test subjects who have toenail infections to see if the relative abundance of the fungi has increased instead of just doing everything in separate little worlds. We do most of our studies as longitudinal studies, and we find that that really also powers our analyses. So this would be our typical analysis pipeline, where we do an initial analysis on the subjects, and then we bring them back after a long period of time, which is like a year, and then a shorter period of time of about a month. That allows us to sort of analyze what is the stability of the microbial communities, and in fact, also these 3 different time points also power our assemblies when we're trying to do metagenomic assemblies. And so here's just 3 examples of healthy volunteers. Like others, I'm sort of showing this as a relative abundance, where the 100% here is 100% of the mapped microbial reads. And so you'll see again, these are 3 different volunteers, and I'm just showing 4 representative body sites. Taking you through, like I've talked about how the chest is an oily site, and that would be dominated by this Propionibacterium. And this would be the person coming in at time point 1, 2, and 3. So the long time point and then the short time point. And what we're seeing is that each person has their own microbial signature. But when we saw it just once, we always wondered, is this just sort of some sort of, microbial bloom or something? So for example, if we look at the side of the nose, this person here, volunteer 2, this taupe color is the Malassezia, or the fungi. And we saw that this person had a lot of Malassezia during their first visit. But you can appreciate still that this person with the taupe colors is really dominated by, has a lot of fungal representation, and that will still be there a year later and still be there a month later. And there's some real outliers, like on the side of the nose, every once in a while, we see people, this is a Propionibacterium phage. So we see this and we think, oh, this person is having some sort of phage bloom. And we think, didn't particularly look anything special. But we come back a year later and it's still all Propionibacterium phage. So there are a lot of elements to the microbiome that if you looked at any of them, you would think, oh, this person is something weird. Well, that's their normal state. And I think what this is coming to for us is that the microbiome analysis is going to be really hard, in that if I tried to do an association study and said, here's 50 healthy individuals and here's 50 individuals with disease, it's really hard to, you have to have something that then the difference between disease and healthy is larger than the inter-individual variation, which can be quite large. But what does power your studies is if you do longitudinal studies, because there is a fair amount of longitudinal stability. So if I power this person on themselves and look at these sort of chronic relapsing diseases, I can actually see the changes pretty with a smaller number of individuals. So while we're talking about these and we sort of get to this relative abundance, we've often really thought about what are the, where are these functions encoded? And we've heard this story told several times at this meeting. Is the function encoded at the genus level, at the species level, at the strain level? Maybe even for some of these bacteria, we've got some interesting stories on that now, the stage. Like, is it when does this bacteria only express this during stationary phase, or does it only express this when it's at growth phase? But we wanted to really try to get to strain level resolution, because we do think that there are a lot of bacterial interactions that are occurring at the strain level. So we're gonna go through P. acnes, Propionibacterium acnes, and Staph epidermidis. These are again just like collapsed views of our 15 healthy volunteers. showing you on the sebaceous sites, there's a predominance of this Propionibacterium, but also showing you that Staph epidermidis, can be a dominant not dominant, but it's present for many of the microbial sites, and sometimes it's just present a little, but that little is still enough for us to do analyses. Okay. So we sequenced hundreds of Staph epidermidis and also pulled the genomes to sequence hundreds of Propionibacterium acnes and made these phylogenetic trees. And so, for example, with the Staph epidermidis, it's a 2.45 megabase genome, so 2,500 genes about. And bacteria can be quite different one from the other. So for a Staph epidermidis, 2,000 genes are core. They're going to be shared amongst all the Staph epi. But the other 500 genes are picked from quite a large pangenome, which can, it can exceed even the size of the core genome. And those can be some of the most interesting genes. They're usually encoded on the chromosome. They can also be on plasmid or on phage. But those genes that are part of the flexible part of the genome can be the toxins, virulence factors, epitopes, antibiotic resistance. And so the skin, we have a low biomass sample, but we have hundreds of these isolate sequence. So we actually have the ability to map our reads back to reference genomes, and we know that with these genomes, we have spanned the pangenome. So Allison, when she was a grad student with me, and in collaboration with Evan Johnson, developed a pipeline called ClinPathoscope, where basically what we're doing is we're reassigning reads that are from the core genome relative to SNPs that we find in the core genome that we map back to the clades, as well as reads that are in the more flexible part of the genome. So imagine if I have reads, and really all I have is strain A, but the reads from the core are going to map to all three of the strains because they're from the core. But what Pathoscope will do is reassign the reads mapped to the core based on SNPs in the core genome, but also based on reads in the pangenome, so that we get a strain level resolution. And there's a lot of simulations that we did to demonstrate this. That's, of course, buried in the supplemental material. And our question was, are the strains present? What strains are present on a human? Is it multiple strains? Is it a single strain? Are those strains specific to the individual? Because I think that's what I'm really trying to get at here. I think what we have is a strain-specific signature for an individual. So here I'm just showing you 3 different healthy volunteers and the Propionibacterium. And you can see that these people have multiple strains of P. acnes. But if you have this strain, these strains on the alar crease, the side of your nose, then you are more likely to have those same strains on every other body site. I'm showing you here just the hypothenar palm and the toe web. So while I've said that the genus and species of bacteria are determined by the body site, the strains are more specific to the individual. You see that here, that between body sites, if you find a strain on one body site, you are much more likely to find that on another body site. With the Staph epidermidis, we see that same model where finding that strain on one body site means that it's much more likely to be present on other body sites. We also do see a tropism for the feet with these brown clade of Staph epidermidis. That it's a separate clade up here, the B clade, which are all the those clades. We rarely find any of these associated with infections. So for Staph epidermidis, we have both a tropism for the feet with the B clade, but we also see the strain specificity. And now also, importantly, I'm showing you again those same individuals, and now I'm showing you it over time. So remember, we brought these people back a year later and a month later. And what I'm showing you is that those strains that are on the individual are retained. So it's like, you know, you wake up in the morning, you take a shower, you do all these things, you know, you go to sleep, you wake up in the next morning, you still have those, I mean, those same strains, and it's going to hold for as long as we've done this analysis. So, you know, the individuals have different strains, but they keep those strains, and even to some extent within the same sort of relative abundance, remembering again that this is over a year and over a month. So why would people have these, you know, what we're seeing is these people have multiple strains, and I'm just showing you here that, again, like this is, you know, 2 healthy volunteers. And with the P. acnes, with my color coding, sorry, I should have mentioned this, that the colors mean that they're more closely related phylogenetically. Healthy volunteer 11 and 12 have different strain combinations. But what we see, you know, we calculated the pangenome when we made that phylogenetic tree. And I told you that, you know, the core of P. acnes has 3,000 genes, but the pangenome is 3,800. But no P. acnes can have all 3,800 genes. They control their genome size. So they can have, you know, 3,000 genes, and then they get to pick like 100 more or 200 more from the pangenome. But they can't take it all. So what we're seeing is that when you have these multiple strains, what you've done is come very close to maximizing the entire pangenome. And so, you know, I think it's really, you know, we all have to think about how our own culture and society maybe does impact our research. And what I think bacteria and microbes are doing in general for me is making me remember that we are communities. And in this sense, you know, maybe what I'm seeing here is that this bacteria doesn't need to hold every gene if it can borrow that gene from another bacteria or borrow that product from another bacteria when it needs it. And that, you know, the bacteria, the selection, and I think this is a direction the field needs to move in, is sort of thinking about community assembly and what the bacteria may be getting from each other. I think that's a major direction that everyone in the field is heading. So I wanted to tell you about 2 clinical stories that we've been working on. The first is a long-standing interest in atopic dermatitis, where kids it's a very common disease, and as I was saying, it's increased in the last 30 years with no change in the diagnostic criteria, which makes us think, is this, you know, something that is a host microbial associated disease? We're very interested in atopic dermatitis because we're hoping that if we get better outcomes for atopic dermatitis, we might also get better outcomes for asthma, allergic rhinitis, you know, sort of chronic diseases. So it's our same study design where we recruit children with atopic dermatitis. And again, it's going to be a longitudinal analysis where we study them at baseline, flare, and post-flare, and compare them with healthy children. And we're sampling them at sites of disease affliction, which is most commonly the antecubital, the bend of the elbow, and behind the knee, and some other sites that we do sometimes see some of the symptoms and some control sites. The disease severity is measured with the score of AD, SCORAD, and what you can see is that at flare, the SCORAD is higher. Then when the kids have treatment, the SCORAD goes down. We'll come back to, which we didn't see at first, but these are actually, you know, this tiny little split here does end up being relevant. What we're seeing in the kids and the healthy controls and at the baseline is that there's a diverse microbial community, again, seen at the relative abundance. In the flare point, what we're seeing is that many of the kids become dominated and have an increased abundance of Staphylococcus when you compare flare to post-flare. For some of the kids, it's an increase in Staph aureus. For some of the kids, it's an increase in Staph epidermidis. And I'll show you that in more detail on the next slide. And again, importantly, this is our model is again where we do microbiome sequencing, but we also are culturing from each of these kids. Okay, so here's the breakdown, where now I'm just showing you on the y-axis the relative abundance of the Staph epidermidis, and the x-axis the relative abundance of Staph aureus. And so we're sort of looking at different body sites in the flare time point, and I'm sort of trying to visually, I'm trying to make a simple figure. Tell me later if you understand it. Yes, but there is a lot of data that's compressed in here where we're trying to show you also how bad the kid's disease was with their objective SCORAD. But what we did was actually, we're laying them out based on their objective SCORAD. So this is the, I mean, this would still be clearly a kid with atopic dermatitis, but we're arranging it based on their SCORAD. And what we saw was that there really are two classes of kids. There's the kids who, in the flare, it's the Staph epidermidis that's increasing, and there's the kids who have a mixed, but that are trending towards having an increase in Staph aureus. And that actually is both associated with SCORAD and also with age. And so we are still following these kids. We're interested to see if a Staph epi kid, we followed them through several flares now. And we see a Staph epi kid will flare with Staph epi on a second, you know, if that's your type, and you'll flare with that, with Staph epi again. But we're still interested to know over a longer period of time if a Staph epi kid might switch to being a Staph aureus kid. We haven't seen that yet. But that might be important in terms of clinical relevance. These Staph epi strains, they're all MRSE, so methicillin resistant. The Staph aureus strains are methicillin sensitive. But I want to go on and talk about Staph aureus a little bit more. So again, these are our 5 kids who, when they flare, they're flaring with Staph aureus. And now I've kind of combined what I told you before about our strain tracking with this. So I've whited out everything that isn't Staph aureus, and I'm showing you that. So you can see that these kids do still carry this same strain of Staph aureus in the, oh, I'm sorry, this is switched. That's a, somehow I was working on my slides. This is the flare, this is the post-flare, sorry. And so we're seeing in the multiple different body sites, but in particular in the sites that are affected by by atopic dermatitis, an increase in the Staph aureus. And interestingly, it seems really to be a single strain that is carried by each child that is present at flare. And interestingly, it's a different strain of Staph aureus that we're seeing for each of the kids. So the strains are matched here to, the reads are mapped here to the phylogenetic tree. But we also then did culture this organism from each of the patients. So we then confirmed that our mapping of these reads is synonymous with then culturing and sequencing the isolate. But we also have these isolates, which then allows us to go into the sort of translational pipeline. So we've got these isolates where we have Staph aureus from the severe flare kids. We have Staph aureus that we got from the nose of a healthy child, and we have Staph aureus that we ordered from, ATCC, which is, what many people would do to take the next step in this experiment. Then we also have as controls the Staph epidermidis strains. And we're using a model that was developed by my colleagues Shruti Naik and Yasmin Belkade, where the mice are colonized these are wild-type C57 black 6 mice. They have intact skin barrier, and you're colonizing them. So it's like as if they're encountering a microbe, that, under a normal state. But we're putting them on the, sort of bathing their little ear in Staph aureus. And what we see is that basically now I'm measuring ear thickness because that's sort of, we also look at immune cell infiltration, but this kind of hyperplasia is a hallmark feature of atopic dermatitis modeled in mice. And so what we're seeing is that if you do these experiments with, well, Yasmin has done it with multiple microbes. We've done it, to show you here. If you use Staph epidermidis, really no effect. If you ordered USA 300, which is an MRSA strain from ATCC, no effect. We're not seeing any effect on the the Staph aureus taken from the healthy child, or when we see it from the less severe AD patients. But the Staph aureus that we're seeing from the more severe AD, this is already initiating features of atopic dermatitis, including this hyperplasia. I would mention this is interesting to us, that this one strain that doesn't seem to increase epidermal thickness or an immune cell infiltration, this is our only Staph aureus strain that we got from a kid who we can tell has a filaggrin mutation. So maybe that's a gene environment, another sort of subtle part of the gene environment interaction. I wanted to highlight that, really talking about where are we going with these kinds of ideas, and really that the field, that this is a rich field. So I wanted to highlight, some of the ideas of my colleague, Rich Gallo, who's, doing similar studies, but he's also looking for commensal staph species that compete with Staph aureus, and has identified lantobiotics from these strains and is now moving forward with probiotics to try to test some of these. I think there's a lot more we could find in terms of the colonization resistance and the community structure. And also, I just thought this was such a beautiful paper from my colleagues, Tiffany Starschmidt and Michael Rosenblum at UCSF, where they're again using these animal models and showing that in early life, when you expose a mouse to Staph epi, you induce tolerance to it, so that if the mouse is challenged with that strain as an adult, it would, sorry, I'm actually sorry. If you expose here, if you expose that mouse, then you can induce tolerance. But if you just expose the mouse as an adult and then re-challenge, you do not induce tolerance. So this actually is, if those of you who follow it, like the LEAP study that was done with peanut allergy, and kind of coming back even to Jack's talks of, if we expose kids to things, they may induce tolerance to it. And it may be that we really should be thinking about, so we all should be thinking about those early years when the immune system is developing together with the microbial communities, and inducing tolerance to some of these microbes. So with our data, I think that's particularly interesting and really cool. I'm thinking about, if we see a single strain of Staph aureus on a kid, could we model that in mice? Could we induce tolerance to that? Could we colonize that mouse with one strain of Staph aureus that maybe then would compete with other strains? What I'm trying to say is basically, we use the genomic data to generate hypotheses that we are then, we generate these hypotheses, and then we are developing the resources between the strains. I think the strains are going to really be crucial, because you can't move DNA from a human onto a mouse, but you can move these strains. And I think the strains are going to be really important, that those strains are from clinically relevant subjects. And so we're using both animal models. We also are really trying to develop the skin culture models. Again, we learn from our colleagues. And so we're really trying to develop these models to then develop therapeutic strategies in the mouse and come back to trying to test these therapeutic modalities in humans. I wanted to talk about another study that we're doing, which is now going to focus more on the eukaryotic viruses. I think this is something that you haven't seen it before in my slides, because it was really hard to characterize what are the viruses on human skin. They were there at very low abundance, and we didn't have very many reference genomes for them. But they occupy a different niche. These are eukaryotic viruses that probably live within human cells. And this sort of combines two interests of mine. which is looking for viruses and also thinking about patients with primary immune deficiencies. And we sort of first started studying these patients because a lot of them do have eczema, but we're also more generally interested in primary immune deficient patients because we talk a lot about you know, how the microbes tune the immune system, but the question for me has been as much, does the immune system really do surveillance of the microbial communities? And you know, we think about our immune systems as these enormous shields, but I think there is a combination where the microbes are probably being selected, yes, by the immune system, but also by the nutrients available on the human skin. And so studying primary immune deficient patients really gives us a window to think about what is the, you know, to what extent is the immune system selecting the microbes. And this was a cohort of patients that are seen at the NIH Clinical Center, which have mutations in dedicator of cytokinesis 8. I'll give you a little bit of understanding about what that gene does in the next slide. But we started studying these patients because of the eczema, but also importantly, they have a lot of viral involvement. So you're seeing here molluscum contagiosum. Most clearly, the vast majority of these patients have these severe warts. We also can see herpes virus infections on some of the patients. And just to sort of give a little background, this is dedicator of cytokinesis 8. And there is a combined immune deficiency in these patients, but there also is an understanding that there might be something, you know, so there's peripheral immunity and systemic immunity, and there might be something specific to the skin of these patients, because the defect in these patients, DOC8 is involved in actin cytoskeletal rearrangements. And what happens in the patients, this is skin, what happens is the immune cells are going to have to migrate through this very collagen-rich dermis. And while normal T cells can do that, the DOC8 patients can't rearrange, you know, can't reorient their cytoskeletons, and a lot of them will just lyse as they're trying, or die as they're trying to go through the dermis. And so we actually think that there is as well a more severe phenotype in the skin, or a different phenotype in the skin than we're seeing with other immune deficiencies. So when we do the skin microbiome sequencing, again, just swabbing their skin, what we're seeing is that, again, you know, I'm showing you here now the kingdom level relative abundance. So for most of these healthy volunteers, it would all be bacteria, or, you know, the vast majority would be bacteria. Then you'd have some fungi, some phage. This is the patient, or the healthy volunteer we've seen with the, like, you know, highest amount of eukaryotic viruses. You're talking about like 8%. Whereas our 27 DOC8 patients, this is aggregate data. We've looked at 8 different body sites, and I've just collapsed it here to give you the overall view. These patients, their skin microbiome is overwhelmingly viruses, eukaryotic viruses. And when we look at the family level, it's a combination where we're seeing a lot of papillomavirus. The pox virus is always going to be molluscum. We see polyomaviruses, but the majority of this is going to be the papillomaviruses. Just to sort of take this off the table, we also did 16S and ITS1 amplicon sequencing. We could kind of get that from the shotgun metagenomics, but I think, as you so nicely pointed out yesterday, sometimes that's not the best method, because we had so few reads from the bacteria and the fungi. Yes, it's concordant, but I felt a little bit better actually confirming it just with 16S and with ITS1 sequencing. That basically their bacterial and fungal communities are relatively similar. The one difference we saw is that the adults' oily skin is more propionibacterium dominated, where we still see a more diverse community. Talking with the clinicians, their skin is also drier, so it's hard to know sometimes whether it's, you know, physiology or really some selection. But in general, I would say what we saw was a fairly normal skin microbial community at the bacteria and fungal levels between the DOC8 and the healthy volunteers. Again, saying that like it's not like we've opened up the niche and now every bacteria or every fungi that's in soil or water is now going to colonize these patients, even though they have a fairly profound immune deficiency. So we were interested in learning more about the viruses that colonize these patients. And so with the shotgun metagenomic reads, we're now going to try to see, can we identify novel viruses from these patients? So we have these reads that we call the dark matter, that don't map to anything. So I've taken away the human reads, the bacterial reads, and now we've got the viral reads and the dark matter. And we use metaspades to do de novo assembly of these reads. And so they'll assemble, in this case, into an HPV genome is sort of 7 to 8 kilobases. We get full HPV genome assemblies from this. And then we can use, sometimes there are things that are already in GenBank, but the novel viruses, we're going to then do further work, which is pretty much the HPV taxonomy is based on the L1 gene, which is the most highly conserved gene in the HPV. And if it's a new genera, species, or type. And from all of our DOC8 subjects, we can now see that we see those same viruses. We can identify 250 new HPVs, which is we have new variants, we have new types, and we have a new HPV species. So this species means that it is less than 70% identical to anything else in the database, but within that new species, we now find 25 new types. So you're seeing that here. So this is our phylogenetic tree of all the HPVs. And anything that is colored in green, we tried to color code it so you can see the HPV genera, because we're finding novel beta HPVs and novel gamma HPVs. And then that color coding, we tried to maintain that here so you can see, we also found a novel mu HPV. So you're seeing like all of these greens are novel beta HPVs. Here you're also seeing it where you can see that the two different colors are, each one of these is a novel HPV species, and then the size of the donut is relative to how many types are within that. So here again is our novel gamma. So now I've messed up the colors, sorry. I think this is something, okay, I've messed up the colors somehow here. This is our new species with all the new types in there. And that does change a lot about how many reads can we map. So again, the lighter blue here are the novel gamma HPVs. So you can see that like for this patient, for patient 1 or patient 2, this percent of the reads that are now mapping to a novel gamma HPV, I would have not mapped them. They would have been in the dark matter for me. So sometimes I've gone from like being able to map 5% of my reads to being able to map 95% of my reads, because these genomes were really just absent from the database. So we find an increasing number of HPVs on both the children and the adults. And we've really, in this case, just expanded the space of understanding of eukaryotic viruses that can colonize human skin. This was done with DNA sequencing. We also went and did RNA sequencing to try to find novel RNA viruses. This was perhaps less rewarding. Because we found a few new viruses, but it's not the sort of hundreds of viruses that we were seeing before. This is in particular sequencing from the nares and the oropharyngeal. We identified some novel rhinoviruses, parainfluenza virus. One of our patients did carry rubella virus, which we then, we got a little worried, but then we typed it and it actually is the vaccine strain. You can see the mutations. But that did suggest to us that patients, this was a foreign patient who could have been exposed to it, but also was vaccinated. So the question was, still, when I say how we try to power these studies with longitudinal sampling. So our question was to look at the temporal stability of these eukaryotic viruses. And so these are two different visits. Actually, the majority of these patients are now going forward for transplant at our center because of a cancer predisposition. And so we grabbed a second baseline right before they started conditioning. So there are some differences where some of the patients were seen 1 month apart. Some of them who had been seen much earlier were getting as much as 5 years apart from the two different sampling times. And this is sort of at the family level. You're seeing the colors of, or at the species level for the HPVs. And what you're seeing is that there is a lot of, there is some chaotic nature of this. It's not as stable as what we're seeing. And I'll take you through some of this data. This is, for example, this patient where you can see had, basically no molluscum and now is having molluscum. And that really did correlate with the clinical situation where you're seeing on the second visit, this is molluscum, which then does come to dominate their microbial community. So the clinical features were also mimicking the sequencing. And we actually looked at this in greater depth, like when we saw warts, how often did we see HPV? When we saw other sort of respiratory viruses, how many times would we find those on the, in the sampling from the oropharyngeal? And actually in these patients, we would always see HPV, whether it was an affected site or an unaffected site. But the levels of HPV would be higher when it was seen with warts. Interestingly, as these patients are going through transplant now, we're seeing some signs. So you further immune suppress them, we're seeing some warts, like even in areas that looked like they were healthy, you see these warts come out when you immune suppress them and then they melt away. The HPVs, our question was, can they clear these HPVs? If they don't have an immune system that's functionally active, can they clear these HPVs? And so this would be, so let me take you through, this is a patient who we could detect 29 types of HPV on their skin. And over the period of 1 month, they lost 5 HPVs and they gained 8 HPVs. And usually what I mean by this is that we're sampling these multiple body sites, so we can see them losing them from both sites. We can see them gaining them in both sites. And we see a fair amount of just sort of juggling around, where even in a month, this patient who had 8 body sites will now, 8 HPV types will now, lose 25 of them. Did I say that right? Has 35 HPV types, will now lose 25 of them and gain 10 more of them. So there does seem to be a fair amount of HPV shuffling around, which again means that probably we are all being exposed to a fair amount of HPV just in the environment, touching doorknobs. So in summary, what I've tried to talk about today are really all of our interests in the many domains of the microbiome. For bacteria, we're really focusing now on how strains really matter functionally and trying to set up systems in our 3D cultures where we do this bioprinting of human skin. Again, it is an advantage of skin. We have had a lot of technology for skin grafts, so we can actually grow skin in incubators and we can print it. And we've really been trying to get at these sort of functional interactions between bacterial strains. as I said, along with our sequencing, we've been doing these deep culture collections, like Trevor, but we're trying to just sort of, we have these Corynebacterium, Actinobacterium, and they're all from the same subject, from the same body site. For fungi, I don't have a story yet to tell you, but I can say my lab has been working for the last year very hard on a new emerging fungal pathogen, Candida auris. One of the first outbreaks was reported here in the cardiothoracic surgery unit in London. We are now having outbreaks with this in the US. This is a fungal pathogen that has evolved resistance to all three classes of antifungals. And I really think this is an area that we need to be paying attention to. There's just not that many drugs for treating fungal infections. And so we're really interested, again, in what are the underlying risk factors in terms of the outbreaks we're seeing in the US are in long-term care facilities, skilled nursing facilities. Is there a relationship with the bacterial community, with the fungi? So that's a big project in the lab now. In terms of the viruses, the DOC8 was sort of just the beginning for us. We're really trying to look now at a lot of different primary immune deficient patients, and also the more common situation is the viral activation for patients as they go through transplant. Some of the aggressive skin cancers that we're seeing in those patients, especially the ones with solid organs, or just the bulk. Some of them develop quite a lot of squamous cell carcinomas. So really what we're looking for is developing each of these projects, but also then really looking at the trans-domain interactions and developing these microbial systems so that we can really have both excellent basic science and excellent translational science to bring forward for clinical benefit. And with that, I tried to give the names and show the faces of the people, but when we're not in the lab, we actually do try to get out and remember that we also live in a beautiful place. And with that, I'll finish and just thank you again for your attention and also for this lovely meeting.
Thank you so much, Julie. That was absolutely fantastic.
Fantastic talk. So you said in terms of the bacteria that the genus and species are specific to a body site, but the strain is specific to the individual. So is there a generational aspect to it? In other words, is there inheritance from presumably mum? And if so, is there any kind of global population structure to it? By that I mean across ethnicities or across regions.
Right, so what I meant to say was that the relative abundance is specific to the body site. So I mean that the oily sites are going to be dominated by the Propionibacterium because they actually utilize the sebum as a food supply. And so so the relative abundance of the Propionibacterium is dependent on the body site, and then the strains are more specific to the individual. In the same ways in which the field is struggling to understand, is there maternal transmission, or really I should just say family transmission? I think those are the same kinds of questions that we want to understand about the skin, whether the dominant strains that you inherit and then stably inherit are from the family members. And that requires a birth cohort. And a birth cohort where kids have been analyzed at the strain level, right? So there's some challenges in that, but that is exactly what we're trying to do and trying to understand. And we are partnering together with people doing birth cohorts to try to get at that. And I think that would be really important, especially in terms of understanding that for the commensal strains, but also maybe understanding if strains of Staph aureus are shared amongst family members, and that's part of the transmission network. But I don't know the answers, and it will take years to find that out.
We've got a question
Yeah, thanks, Julie. That was really great. I had a question with respect to the data that you showed for Staph aureus and Staph epidermis in relation to within population level genetic heterogeneity. And if you see between both species at the strain level that there would be differences in terms of the level of heterogeneity, and if this might be somewhat related to different lifestyles in comparing, for example, more specialists versus generalists, and if thereby perturbing the skin, one could, select for one over the other.
Right. I mean, that has been an intriguing question in the lab. Sort of, for commensals that you're seeing this multiphyletic, multiple strains, but for Staph aureus that we're seeing a single strain dominating. And again, asking these questions, which I think we would need to then model, is it that there hasn't been exposure to multiple strains of Staph aureus? I sort of doubt that, but rather that there is a different selective force on a pathogen than on a commensal. And then those are the kinds of things that I think we need these sort of complex communities to understand, especially because, Pear's Bork study, Simone Lee, and then, again this morning, the work that you presented, beautiful talk, no, not Naso, sorry, but, about the FMT, where if you have one strain that opens the niche, is that true? Is that going to be true across all species? Because, even coming back to the earliest work that S.aureus did, where sometimes having one strain opens the niche for other strains, but sometimes in the gut, he was seeing there, those are gnotobiotic mice, that one strain can compete with other strains, and you get this, strain level resistance. I think there's so much we have to understand from that, and even like S.aureus's data, then with Romnick's birth cohort, the strains that, S.aureus found in the gnotobiotic mice that could compete were different than the radiation that Moran and Romnick were seeing in the birth cohort. So yeah, everything we need to understand. And I'm sorry, I didn't ask your question about geography and ethnicity, but that's because all of our studies are too small. They're too underpowered to really get at, is there any sort of level of organization based on race, ethnicity?
Very nice, Julie, as usual. With the strains being the same within individual, can you see signs of adaptation to the different body sites and even go down to the molecular underpinnings of that adaptation when you use the data across individuals? Then?
Yeah. I don't think we don't have that kind of resolution. Again, I think that we'd want to model that, like colonize mice and then take the strains off the mice and see if there was microevolution or adaptation. Because in humans, you never know, was that originally there and got selected for, or was that an adaptation? I don't feel like we'd be able to sequence deeply enough to do that, but those are really good experiments, but I think those would have to be done experimentally, because it would be hard to convince me as a reviewer that you had sequenced enough to distinguish between this was a rare species and this was an, in vitro or in vivo evolution.
Okay, one last question.
Fantastic presentation, thank you. Just one quick question. Do you have an opinion or some comments on the impact of the gut microbiome in impacting the skin? Because I understand at times the gut is impacting the liver function and causing problems at the skin level. So any thoughts on that?
Yeah, I mean, probably Shruti did that work early on, and we see them pretty much as two separate compartments. But I guess I would always caution that when you look at germ-free mice, that's another thing. Probably those microbes are colonizing the skin. And probably causing effects there too, because they're really not native species for the skin. But in general, we don't see, I think there is this triad now, which used to be atopic dermatitis, asthma, allergic rhinitis. People are now including food allergies in that. So I think that, all of this is localized but also can become systemic. But there's not a strong connection. It's not like the oral cavity and the GI tract.